Okay, thank you very much. My name is Sami Datwani. A little bit about my background. I have a doctorate in chemical and biomolecular engineering from Johns Hopkins University, and I'm going to try to convince you that the future of science is sound. Going back to the grand keynote this morning, the focus of this is in the digital health space, so we'll be sort of switching gears. So I thought I'd start with a quote. To raise new questions, new possibilities, to regard old problems from a new angle requires creative imagination and marks real advance in science. And I'm going to try to convince you that's what we're doing. So essentially, we have a platform that is aimed at revolutionizing liquid handling, but providing a solution. And the idea is that with a revolution, we have a sudden, radical, or complete change. And literally, the way we do our solution, we are turning the world upside down. We are scaling down. We're using a new method of liquid handling that is giving us some far-reaching advances in science. We're using sound energy to transfer quantized or discretized amounts of fluids, a billionth of a liter, with extreme precision and accuracy, and that's going to change healthcare. We don't use pipette tips, and that's a paradigm shift, and we actually see that there are better results, and I'll show you some of that. And we think that acoustic droplet ejection is becoming critical and an all-purpose tool for life sciences. So Labcyte, let me just tell you, is a small startup in the Sunnyvale area. We've been around for a few years, and we're starting to gain some traction. So what's the disruptive technology? Basically, we have digital images of dispensing a drop. There's some photomicrographs, and essentially we are working with nanoliter volumes. So we're scaling down the lab. The way we do that, it's very accurate, it's very precise, and these increments preserve sample integrity. We transfer to an inverted surface, you can see from the photomicrograph, so we turn the world upside down. Our destination plate coming from our source plate is upside down because of the small volumes and surface tension. The experimental quantities, the biochemical assays, they all just stick. We are doing a lot of signal processing in the background, so we're adjusting in real time. We're calibrated for differences in evaporation, hydration, changes in fluid properties, surface tension, viscosity. So we eliminate the need for the user to do that. And this touchless paradigm essentially eliminates tips from the process, and that is where we get a lot of error. So here's a closer look of the instrument. We call it the Echo. Bats use echolocation, so we're using similar features. We use sonar, sound navigation and ranging, and ultrasound is the range that we're working in. Essentially, we deionize the source and destination stages. We transfer upside down. You can see the piezoelectric transducer generating sound waves. We generate more volume for the assay by dispensing many times, and we can essentially access any well and move to any well on a plate, so we're not fixed by pipette heads. So there's no calibration, and we don't use tips. We have the software feature that enables that, and that is essentially taking what's in my head and understanding the features of a fluid and managing that in real time. So we adjust to the real-time fluid properties. We're able to handle a wide range of fluids from cell lysates, various reagents, enzymes stored in glycerol, DNA, RNA. There's no operator intervention. And we miniaturize these processes, so we are really in a high-throughput environment. And this is what's going to generate a lot of the data. So how do we revolutionize life sciences? Well, most of what I'm going to talk about are what customers are saying, and I'm going to talk a little bit more about the areas we're going into. So next-gen sequencing, mass spectrometry. We can transfer single cells, so we've enabled that. And then we have this foundational technology that we can apply in a variety of areas. So we help to identify new drug candidates. We help to accelerate drug development. We repurpose drugs. We have a system for truly personalized medicine, gene editing, and then low-cost diagnostics. So all of these are achieved, and they're in publications that I'll show you at the end. We are able to get away from the problems with traditional systems. The problems simply are human error, liquid sticking to tips, viscosity reducing accuracy, surface tension reducing accuracy as well. And essentially our technology eliminates these top 6 sources of errors. So the Labcyte system. Essentially the heart of the system is this Echo liquid handler you see in the middle. We have wraparound automation. You can think of the robotics that you see in the semiconductor industry. We've had to write software to enable us to give these capabilities. So cherry picking, going back to the same well, doing some things like dose response. All of those are enabled by our software. And because we have to hear through the plastic, we've now got into the consumable business. So we're making our own source plates, and that's what works with our platform. So altogether, this is the solution. A couple of quick applications. The first one is kind of interesting. On the left, you can see it. This is pulled from a patent directly from the literature where a customer, AstraZeneca, basically ran through something called an IC50 where they're doing a dose response to determine the efficacy of a drug. They used a competitor device and they used our device, and they got totally different results. Turns out they would have missed this drug compound because of this, and using our technology, they're able to highlight that and then take that drug through development. Another example is some folks at the Finnish Institute of Molecular Medicine have been using our technology, and if you think of cancer, it's a personal thing. Everybody has different genes, things react differently. So what they're doing at FIM is pretty interesting. They're taking cancer cells from the patient, they're taking those cells, growing them in our plates, dosing them with different therapies, different concentrations, and they have all the flexibility with our technology. They're lysing the cells, and they're running qPCR and sequencing beyond that. And they're finding out that they can now go back to the patient and say, this drug is going to work for you. And we have examples with patients that have an AML patient that supposedly was given 6 months to live, that's still alive 6 years later. So this is a quote from Olli Kolmeni, who basically says that the research opportunity is changing the way new drugs are introduced for patients. It's a game-changing, and Labcyte technology is a key platform. I'm going to talk really quickly about new technology we're working on. It's coupling our acoustic technology to a mass spectrometer. Mass spec is essentially the gold standard, the holy grail for biochemical analysis, and we're coupling that to enable high throughput, low cost, label-free analysis. So essentially, we put our technology in front of the mass spectrometer, and we're able to go higher throughput and compete. So our throughput is roughly 3 to 30 times faster than the current technology that's out there, and it's really quite a game changer. Probably the one thing I'll highlight is running through sample screens. We can do that in hours for millions of compounds, whereas competitors would take days. So there's savings from that, and we're working with some partners, AstraZeneca and Waters. The best compliment is when people write about your technology, and this graph here shows you cumulative almost 400 scientific publications in a variety of areas. So we're continuing to grow in that area, and here are a couple of quotes from different journals: Nature, Drug Discovery Today, the Journal of Lab Automation, highlighting how researchers are using our technology and advancing their research and science. We're also in the synthetic biology space. I think one point I'd pull out is the cost per sample decrease from on the order of $70 to 70 cents. So we're really trying to change the way things work, and I'll leave you with some of our customers. Thank you.